Boys and girls, it's the weather report for Friday, September 2nd, 2022. And oh, my butt fungus. We're putting the butt fungus link out again?
That's what I think we should do.
We should put the butt fungus link out.
It's a Joel Duff video, so it's going to be good. It's going to be scientific. It's going to be informative.
Yes. And I think Andrew Cumming is upset about something Cell did. No, I'm not sure. Something about, we kind of predicted this whole thing about, the panic over the Big Bang, right?
Yeah, it's been going viral a little bit.
Right.
At least here in the US, at least. I don't know about other countries, to be honest.
Yeah. And you saw that thing on Maddox's channel where
yeah.
Sunflower or Sundrop or whatever the fuck his name is.
What you mean, bubblegum?
Oh, bubblegum. Yeah. Okay. He was doubling down on that it was Lerner that wrote the panic article, right. I think, or the panic paper or something.
Yeah, which of course he was wrong, but
yeah,
it was just funny because we'll get into this later. But LPP was saying one thing that was going against what the point Sal was making. Otangelo was making a completely different point, so it was kind of a big stew swirled together, and it was pretty entertaining.
Right. So Otangelo came.
Speaking of Otangelo.
Yeah, speaking of Otangelo, boy, are you buzzing tonight? Let's see. Check your audio. Oh, your audio is horrible.
It's a little low.
The buzz is gone, but say something. Do an audio check.
How about now?
I can barely hear you. I can barely understand you. Really bad.
It's better now?
No, it's still bad.
How about now? Is it better now?
Yeah, that's better. But you're kind of weak. I'm gonna have to turn you up a little bit.
I'll turn him up a little bit.
Yeah. So okay. So I was reading that email you sent earlier. Go ahead and say something again.
Is it better now, my sound?
There we go.
Yep, I got you.
A bit better.
I got you up. Okay. I had to turn you up a little bit. But I was looking at that email that Top Dog, and do you mind if I share that?
Yeah, you can share it.
Okay. Let's see if I can put it up here. Get rid of myself and put that.
Sorry.
Okay. Let's see. My question about, so you were talking about biodiversity and complexity, and that would be like the development of limbs and whatever. The best weapon against inferences based on deceptive thought is first having solid facts. There's the evidence. What is what we observe in nature? We have to make the best out of it. And you kind of got it right, like as far as historical, basically what you would call historical science, we just call it science. Trying to figure out what happened in the past that led to, how common ancestors diverged, that sort of thing. That you have to, and I forget how you said it, but you said it somewhere down here. Let's see. That is because, no, I'm not sure where you said it. You said it kind of right, that basically we observe what is going on now. We figure out what can happen. We figure out, basically how genetics works, how inheritance works, all of that stuff, things that we can observe in extant organisms, right? And then from that, we make inferences about common ancestry. And that is the game, and that's how we do it. Okay?
Well, you presuppose common ancestry. I would just say
in
general sense, that based on what we observe, we can try to understand how the things that we observe came to be in the past, what mechanisms that
Okay,
were.
there's no
That's what we're doing.
there's no presupposition at all. Okay, common ancestry is an hypothesis. Okay, you don't call it presupposition. That's not what it is. You don't just say, "Oh, we know this happened this way," right? It's a hypothesis, and then you go out and you find evidence for that hypothesis. So and then you find mechanisms about how that could have happened in the past. And when you have actual evidence of mechanisms and you have evidence for your hypothesis, like we do with phylogenies and genetic similarities and things like that, that's evidence for our hypothesis. And then when we find the mechanism, well, then we put it all together and we have a theory. And that is the theory of evolution, and that's what we've done. But it kind of drove me nuts here when you said you quoted Gerd Muller in his paper. Let's see, what is that paper? It's not the paper. I got the paper here somewhere. Why an extended evolutionary synthesis is necessary. So you quote Gerd Muller, right?
Right.
Let's see. What part do you quote that he says? Let's see. Several of the cornerstones of the traditional evolutionary framework need to be revised and new components incorporated into a common theoretical structure. Okay. But then you go down and say, then he says, let's see, I disagree with the last. Okay. He said instead of chance variation and DNA composition, evolving developmental interactions account for the specificities of phenotypic construction. So you disagree with the last sentence. In fact, you disagree with the entire paper, yet you pick a quote from this paper again. What do we call that, Otangelo? It's called cherry picking. Okay. Gerd Muller wrote an entire paper where he said, okay, we have to make sure that we've completely abandoned the simplistic ideas we had from the 1950s and 60s, 1930s and 1920s, and we have to bring in this extended evolution. And let's see, where does Muller do that? This is it. This is the paper. So right here, he shows basically what his model is for the extended synthesis, which involves morphogenesis or embryology, biophysics, topology, and function, which is really important, and then DNA, self-assembly, cis-regulation, etc. And here we have biophysics of cells, autonomous behavior, and self-organization. So that is the extended synthesis. So you brought up this paper, and the paper is definitely completely counters your views. And I want to know why you keep doing that.
Okay, so what I basically agree with Muller is that there are more than genetic mechanisms involved in the construction and adaptation of complex organisms.
We all know that.
Well, I mean, I have several times linked my paper where I give my opinion on what I think are the mechanisms involved. And what I constantly point out and say is that it is not genetic information, but in a special and as well epigenetic information and signaling networks, which work in an integrated systems fashion together to construct complex organisms.
And we all know that. We all know that.
Okay, so that is today what has to be explained, how that complexity came to be in the past. Now, of course, you have here two possible explanations. You can always adapt and say, okay, it was evolution that brought these things into existence. And this is what we basically disagree on. This is the philosophical circle around the evidence, which we can debate, and each one of us can have a different opinion and come to different conclusions.
But it's not a philosophical problem. It's a problem of evidence. And we have the evidence for all of these mechanisms, and including epigenetics, including basically the 4D nucleome. All of that stuff is part of the theory of evolution and part of the extended theory, right?
No, I disagree with that. These things, they explain how organisms develop, how they are formed. That's
Right.
what it is. But this is a separate thing to explain how these things actually came to be. So, for example, we have the gene regulatory network. We know that it is involved in expressing genes in the sequence, in the time span, how it develops, everything about that. But then a separate question is, how did the gene regulatory network develop or evolve, or how did it come to be? That is a separate question. And
Okay.
there, of course, Muller, he puts always evolution, okay, and that's the thing that I disagree with. What I don't disagree, and I think it is very pertinent and important to be outlined, that's why I bring up always that paper, is that the evolutionary gene-centric view has to be expanded, and other newly discovered mechanisms like epigenetic languages and codes and signaling networks also have to be brought into the picture.
What do you think we're doing? Who do you think is ignoring these things?
Many are ignoring that because they don't even know that these things are involved in
What is your
electoral
evidence that many evolutionary biologists are ignoring gene regulatory networks and epigenetics and all of that? What is your evidence for that?
Well, Davidson himself, which was a pioneer in regards of the gene regulatory network, he pointed it out that the origin of gene regulation also has to be brought into the explanatory toolkit of origins, and many have not considered this, and many still don't do it today. I see it in my conversations with atheists all the time.
Okay, do you see it here? Because you said they're two separate questions, and they are not. Okay, the way a gene regulatory network works today, what we observe, and the way they develop with embryogenesis and biophysics. Okay, those things are how the gene regulatory network came to be.
Wait a minute.
No,
You
hang
are
on.
again.
No, hold on. I'm not done.
Yeah.
Okay, the very same things that tell us how embryos develop, right? And how these, and basically they go through phases where they shut off banks of genes or cassettes of genes. Okay, and we know that. So we know what shuts them off. But anyway, all of that goes into the origin of these gene regulatory networks. Now, when we look at a protein, a particular protein, okay, pick a protein, go out and look at the interactome of that protein. And what you're going to find is that about 5 of those connections we understand go into the gene regulatory network, right? But there are sometimes as many as 100 other connections that protein has. Okay, so we have all of these proteins in this vast network that are kicking around, and a lot of the networks are dead ends. Okay, and that is how it developed through natural selection, purifying selection, which cuts out all the bad connections. Those, it's almost like if you imagine if you've got like an object and you got these little fingers or filopodia, right, that are reaching out and touching it. Okay, they're finding the shape of the object, one little tiny step at a time, until they completely grab that object and they envelop it. So the template basically locks in over time by very, very small mutational changes, duplications, deletions, insertions, all of that. Okay. And that's how gene regulatory networks develop. So the very same science that tells you how they work, okay, combined with the science that tells you what is going on in that interactome, is the source of evolution. And we all study that, and we all know this. We all know this. You're not telling us anything new. The reason the extended synthesis, the reason people get mad at Gerd Muller and the other extended synthesis people, is because, like Dr. Dan says, we already knew that. We're already working on this. Okay. Anyway, go ahead, Andy. But Otangelo, I want to warn you that we're going to have a short discussion here, and then Andrew wants to address something different.
Or I was saying we could do that, or I actually want to ask Otangelo a few things kind of related to that later.
No, Andrew wants a piece of you. Okay, go ahead. Otangelo, did you have a response?
Well, I obviously disagree with your inference, and
Of course.
development of an organism, it is separate to the origin of the development process. These are two different topics. Well,
Why?
because they are not related. You have an organism which starts with a zygote, and if after a certain period of time, it follows an orchestrated process to become a multicellular organism. And if you start with the origin of life, where you have a unicellular organism, obviously that trajectory wasn't there yet. So it had to originate supposedly after life started. So you have to explain how that process actually of development came to be. Now, Evo tries to explain that that process through evolutionary mechanisms, but far from what you claim, I completely disagree that natural selection, genetic drift, and gene flow, the standard answers that they actually have demonstrated to be in place to explain that development. I disagree with that. I've never seen or read any science paper which has demonstrated that to be factual.
Maybe you're Googling the wrong thing. Because
if you have a paper, then you can send it through email and or outline like you do here in the screen. I'll be happy to look it up.
I have about 1,000 of them, but
yeah, send me the principal ones, which are the best.
Okay, now here's the problem. Okay, you're looking at an extant organism. Okay, you got a zygote, right, and it develops into an extant organism, a fully functioning elephant. Okay, we'll pick an elephant, right? Okay, so we've got that. So you're looking at how that came to be, how the elephant came to be. Okay, that's not the right way. That's not the right thing to look at. Okay, you have to go back. Now, the hypothesis is that there was a common ancestor, that there was something simpler that broke off before, prior to the elephant. Okay. And then if we go back further, okay, if you're starting to look at gene regulatory networks and basically these basic body plans, we're going back 700 million years. Okay. And we're going back to very simple organisms. We're now dealing with things like sponges and things that crawl around on the ocean floor. So now, those are the things we have to look at. And you look at how one thing gets to be another thing. And when you do that, what you look at is embryogenesis. That's what Evo-Devo is about. You start looking at how these things work now and what happens if there's a birth defect, if something doesn't work, if we knock out a gene or we knock out a little piece of that, okay? And we end up with something entirely different. But you go backwards and you start looking at the simple beginnings. Like if we're looking at a limb, development of a tetrapod limb, right? Like my arm or the elephant's leg, okay? You go back and you go all the way back to where you have like a long worm and it has a very tiny bud that sticks out of the side of the worm, right? That's where it starts. So we go from that all the way through fins that swim around in the water to things that basically push them up off the bottom to things where they can move back and forth and come up with a gait and start walking. And now we're looking at tetrapods, and then you come all the way up, and then we'll get to the elephant. But you don't look at the elephant and say, oh, the elephant is improbable, because it is improbable, Otangelo. That an elephant would just suddenly appear is very improbable, and no one believes that, except for you creationists, okay? We go all the way back to the beginning. So every piece of that Evo-Devo that we're looking at, every one of those gene regulatory networks are a pathway. Okay, and we look for evidence of how one pathway can turn into another pathway without killing the organism. So we're going all the way back and we're creating a very long story. Okay, your job is to prove that that's absolutely impossible without a God. That's not my job. Anyway, Andrew wants a piece of you. So.
Well, if he wants to give it a response first, and I'll get into it.
Oh, but then I'll have to respond, and we don't want that. Go ahead, Otangelo.
Yeah, I can quote you here. I actually think I have done that before on your channel. It is a paper of Davidson, of Eric Davidson, Evolutionary Bioscience as Regulatory Systems Biology. And he says here very clearly that Davidson is refuting Darwinian evolution with these words: Neo-Darwinian evolution is uniformitarian in that it assumes that all processes work in the same way, so that evolution of enzymes or flower colors can be used as current proxies for study of evolution of the body plan. It erroneously assumes that change in protein coding sequence is the basic cause of change in development program. And it erroneously assumes that evolutionary change in body plan morphology occurs by continuous process. All these assumptions are basically counterfactual. So he is addressing.
So you're making two assumptions there, Otangelo.
Let me quick finish. What he is addressing here is precisely what I said previously, that there was a broad claim or assertion that just genetic sequences were responsible to explain organismal complexity, and he outlines that the gene regulatory network has a decisive role, but not only. I have 17 different mechanisms listed which are actually involved in constructing complex organisms. Yeah, go ahead.
So you're conflating a few things there, Otangelo. You're conflating Darwinian evolution with the modern evolutionary theory. You're conflating protein coding genes with all genetic sequences. Because we know that those things aren't all that is involved, as Speed said just a moment ago.
I am addressing something which maybe you know, maybe Speed knows, but many people and many atheists and proponents of evolution, which I have interacted with in the past and still today, they hold to the view that just genetic change explains.
Otangelo, no offense, I don't honestly care what random YouTubers or atheists on YouTube say about evolution. Right. I care about what evolutionary biologists say about it and what the data shows.
Well, Davidson, he was addressing actually his colleagues and not layman people.
It's kind of cool that you're quoting from Evolution News here, Eric Davidson by Casey Luskin, basically.
I was quoting his own paper.
A cherry pick quote out of one of his papers where he's making a case about the complexities of figuring out how gene regulatory networks evolved. And Davidson passed away, didn't he? I think we lost him.
Yeah, he died.
Wow.
He famously developed a notation and a wonderful laboratory for laying out gene regulatory networks, and he did a lot of work with it. And I don't know, there was something that he came up with. Let's see.
Because just by the context of the quote you gave, Otangelo, it seems he's arguing for a broader picture than just natural selection and mutations to protein coding genes, which we already knew that was necessary in order to explain some of these phenomena, like how development occurs.
Well, my thing is that I say all the time that, I mean, if you know it, if Mike knows it, great. But the more you have integrated complex codes and languages on an epigenetic level, the more difficult it becomes to explain all this complexity just by non-intelligent mechanisms, because they are not just more complex, but they are integrated, and you need to integrate them. that is a big problem.
No, that's exactly the opposite of a problem for evolution. The fact that these things are so interactive and there's so much crosstalk going between these gene regulatory networks is exactly how they evolved.
It's messy. It's inherently messy.
Yeah, Davidson himself, he says that if you actually change just a little bit in the gene regulatory network of an organism, then immediately it breaks down.
Yeah, and when it breaks
Obviously-
down, purifying selection kills it, so that pathway is not going to make it. So the only one that's going to make it is the one that doesn't break down. That little connection in the middle.
It also
Yeah, first
depends
you
on
have-
the context there. Are you talking about like a Hox gene? Are you talking about a lower sort of tier gene on the regulatory network there? Like it isn't just a one shot, one kill everything.
You know, we are talking here about basically two gene regulation systems, one in unicellular organisms like in bacteria, and the other which is more complex in multicellular organisms. And the
What I
question,
said
of
applies
course,
to
is,
both.
and the question, of course, is that let's suppose you have a multicellular organism with, let's suppose, 20, 50, or 100 different kind of cells, then obviously you need to regulate their expression. They need to go at to the right place where they belong in the body, and all this has to be directed through information. And the question is, how do you get a gene regulatory system if you don't have the different cells that have to be directed and expressed, and you have to have the right cell, a neuron in the brain, and the muscle cell in muscles,
Because you don't.
and so forth. So
That's not
in
how
my
development
understanding, you
works.
need both. You need different cells, and then immediately as well the gene regulation which directs that different cell to the right place.
Okay, that's where you've got the chicken and the egg thing in your head, and you can't let go of it. Okay, there's basically for a regulatory network to form in a genome and to basically be end up in a functioning living organism, it does not necessarily have to connect to anything. It can just be a throwaway transcription, translation of a protein or a network or something that goes nowhere, basically.
I'm sure you're familiar, Otanto, also with enhancer regions and how they're a lot more modular than you may think they are. Like there's research
Let
that's
me
been
just
done
quote
on
here.
changing one of those, and it doesn't change the entire organism or all places
Yeah,
where that gene is expressed.
Yeah, let me quick quote here from Davidson of what I just said. No
Okay,
subcircuit function.
let's bring the whole paper up. Let's not look at Casey Luskin's cherry pick. Okay, and I'm trying to bring the paper up, but my drive isn't working for some reason. Where I keep all my paper.
Just a minute, and I'll give you the link to the paper here. It's Evolutionary Bioscience as Regulatory Systems. I'll give you the link.
I'll find it before you do.
Oh, here it is. I got it.
It's a race. We're off.
Okay, here we go.
Okay.
Let's see.
Here's the link in the private chat.
Yeah, I got to go and pull the paper. I've got it somewhere, and I have it highlighted. And that bothers me because I can't see my highlights. Damn, what is wrong with my drive? No, cat, no. I don't need you right now. I don't love you that much. Oh, I guess I do love you, but not now. Okay, 2011. Let's see. Where are we here? We're at, okay, this is it. So what point is he making in this paper? Let's see here. Let me get it front and center. And of course, I can't read it. At present, several entirely different explanatory approach mechanisms by which animal body plans have evolved. The respective relevance is briefly considered in the light of modern knowledge of genomes. I'm going to put this in front of me so I can read it easier. And regulatory processes by which development is controlled. Just as development is a system property of the regulatory genome, causal explanation of evolutionary change and developmental process must be considered at a system level. Here I enumerate some mechanistic consequences that follow from the conclusion that evolution of a body plan has occurred by alteration of the structure of gene regulatory networks. The hierarchy and multiple additional design features of these networks act to produce Boolean regulatory state specification functions at upstream phases of development of the body plan. Okay, did you hear that part? Upstream phases of development of the body plan. These are created by the logic outputs of network subcircuits, and in modern animals, these outputs are impervious to continuous adaptive variation, unlike genes operating more peripherally in the network. So that's what the paper is about. Okay, so he's talking about what? What is your view of what this paper is?
I'll quote here from the paper. No sub-circuit functions are redundant with another, and that is why there is always an observable consequence if the gene regulatory network subcircuit is interrupted. Since these consequences are always catastrophically bad, flexibility is minimal, and since the subcircuits are all interconnected, the whole network partakes of the quality that there is only one way for things to work. And indeed, the embryos of each species develop in only one way. So for me, this means it is just, there is just a very narrow way upon which you can have that regulation. And if you are tinkering, then you have always a catastrophe. So do you think there were 100 catastrophes of tinkering and then suddenly maybe you got one which was functional?
There were probably thousands. I mean, one particular prokaryote, for instance. Well, let's go a little bit higher. Let's look at plants. Okay, how many spores does a plant put out? How many spores are there? So how many of those are going to work and how many aren't? Okay, we have hundreds of thousands or millions of spores being put out by the plant on every generation. Okay, and natural selection is going to take out the ones that mess with the gene regulatory network that is required for a viable organism. But they're not going to mess with the ones that are neutral. The ones that are neutral are not going to have an effect. They're going to be out there. They're going to copy from one genome to the other.
And I strongly disagree with what was said there about the gene regulatory networks being this integrated, because like he even mentioned earlier, I think you quoted that you can tweak more so the genes on the periphery of this network. And we've seen that you can do that and it doesn't have a negative effect on the organism. In fact, some of these types of mutations have actually benefited different populations of organisms. An example that I'm most familiar with is ocean versus freshwater stickleback fish that have been studied by David Kingsley and colleagues. And you just change one enhancer there. The gene itself is unaffected. Everywhere else it's expressed is unaffected, except that one particular region of the body. And you have a fish with spines to a fish with no spines. And that's it. That's all you need for evolution to happen.
Yeah, so what is the upshot?
Well, I compare a gene regulatory network to a library where you have, let's suppose, 1,000 different books, and each book contains the instructions to make a machine, and you need to have a sequence where you make each protein or machine and then connect it to other parts of a larger system or a factory. And that sequence, it must be correct, and then the parts must be interconnected as well in a correct order together in order to become a functional system. And claiming that just non-goal-oriented mechanisms without foresight would be sufficient to instantiate such a system, at least for me, that is a far stretch.
Well, I think that's just where we're gonna have to go on this point, because you're saying one thing, we're saying the complete opposite, and it seems like you think the genome is a lot more non-flexible than it really is. But anyways,
Yeah, you lost me at books because it's not a book, it's a cell. And it's not a machine, it's a cell.
Yeah, but you can compare a gene to a book where you find instructions to make something.
No, you can't. No, you can't. It takes a designer to make a machine. These aren't machines. Okay.
Well, proteins are not machines, Mike.
No, they're not.
Have you ever heard the term molecular machines?
I've heard that term, yeah, and I disagree with it. It's a bad way to look at it. It's not a machine. It doesn't have gears. It doesn't fit together. It wasn't ground and polished and made perfect to fit one thing into another. They don't fit one thing into another at all. They manage to glob onto each other magnetically at some varying strength. That's what they do. They're not machines. They're big globs of molecules and atoms.
One dissimilarity between a gene and a book is either a specific letter is there or it isn't there. There are only two states available.
Right.
A gene can be expressed in increasing or decreasing relative to the wild type out there. So genes aren't the same as books. Like I said, they're a lot more flexible than you think they are.
Well, for me, I just use that analogy to say, in the same sense as you can find instructional information to make a machine, for example, in a book, the same you find in a gene where you have the instructional assembly information.
Well, that's why I'm saying it's not the same. And if your analogy doesn't work, then your analogy is irrelevant.
Right.
Well, so what is the function of DNA?
And an analogy, Otangelo, is how you teach something or how you explain something. It's not how you study something and figure out how something works. Because the analogy at some point is going to break down because it's just an analogy. You're making an analogy. So
I'm making an analogy, but what is the function of DNA?
What is the function of DNA?
What is the function of DNA?
Well, that's kind of a big loopy question that would take me a half an hour to answer. But
no, absolutely not. I answer it for you. It is to store instructional assembly information to make proteins or microRNAs or regulatory RNAs or ribosomal RNAs. But in any case, if we are just focusing to in the human genome, at least 1.5%, it is the information to express the information to make proteins. Now, in regards of what you said of machines that you disagree, well, I understand the machine as a device which is made of several subunits which are interlocked, work in an integrated fashion together to provide a function or to make something which has a purpose or function, and it uses energy. And that is precisely and analogously to what we see in biochemistry, where the protein is a thing made of several interlocked parts which work together. They are driven by ATP energy, and they provide a specific function in the cell. And I think that the term molecular machine is very appropriated.
Put a period in it. Okay. No.
So, Otangelo, would you consider a book to be a machine then? Because you have to have the pages in a particular order. You have to have the sheets. I don't know how they tie the book pages together, but you have to have these in a particular arrangement in order to have a fully functioning book. Would you consider that a machine then?
No, I wasn't saying that DNA is a machine. I said proteins are a machine. What I am saying is
That's
that
not the
a
question I
gene.
asked, though.
Well, but I think you asked a question which was completely disconnected to what I said.
Well, I'm just trying to probe your logic here, because it seems like if you can define a machine that way, then you can pretty much define anything to be a machine.
Yeah.
It's very loose.
And
No, certainly
okay,
not. I brought three different things as characterization. One is integrated parts which work together, driven by energy that make something which has a function.
okay, when you design a machine, Otangelo, does the machine then just assemble itself like a zygote does?
Well, the thing is, if I,
No,
as a machine
answer the
designer,
question.
would be able
No, answer my
make-
question. Answer my question. Does your blueprint just assemble into a machine?
no, you need information transfer,
Okay,
which adds complexity to the whole picture, right?
it does. But anyway, blueprints don't turn into machines, and DNA actually doesn't turn into organisms. So there's a
No,
little
of course not. That's not what I said.
more to DNA than that.
Well, that's not what I said. So
Okay. Not a machine. Not a machine. Cell is not a machine. Machines don't.
no, a cell, no. I said proteins are machines, and I always say that cells are chemical factories.
Cells are chemical factories. Okay, so they make chemicals and then they deliver the chemicals to the supermarket, or what?
No, they are basically self-replicating, and in order to do so, they make the building blocks, amino acids, the energy, the proteins, and then they replicate the DNA itself, the information.
So if we had a factory that did this, the factory basically would start from one brick, and then it would with the information inside of that brick, it would start assembling itself, and it would create a complete factory. Okay, and then the factory would produce what?
It could eventually, that would be the epitome of technology if we as intelligent humans would be able to make self-replicating factories. That has never been done.
Yeah, right. It hasn't. But life is not a factory. Okay, it's not producing anything.
It produces more of itself.
More of itself. That's it. Over and over again.
Yeah.
And then it changes at every step.
Yes.
Every time it produces itself, it makes it a little bit different. So it's not a factory.
Well, making means fabricare. Fabricare is Latin and means factory. It fabricates more of itself.
Okay, so it's a factory that makes itself. Do we have any factories that make? So why are you using factory as an analogy if factories don't reproduce themselves? Factories produce products for us. We build factories to basically, and we people them with, we put people in there and machines in there, and we produce some product. Okay, life doesn't do that. Life just produces more of itself, but
So it
not
produces.
exactly itself, a little bit
It
different.
produces. Yeah, but it still produces, right? It is making
I actually
something.
kind of like what Icarus put in the side chat here. The sun is a chemical factory, because you could almost use the exact same criteria you put earlier, Otangelo, to describe the sun as a machine or a factory. It's driven by nuclear energy. It can make more of itself over long time periods, of course. It's a stable system. So your analogy kind of breaks down if you tug at it at certain points.
Right.
Well, if I can answer to that, in order to make a factory, first of all, you need to know what do you want to make a factory for? What is the product that you want to make through that factory, right? Once you have that goal in mind, you sit on the table and you draw all the specifications of that factory, all the machines that it requires, the materials, the energy supply, how the materials are brought into the factory. Once the product is made, how it gets out in regards of the waste product, how it is disposed. And that is analogous to what we see in a living cell. And
no. Where's your evidence? Okay, where's your evidence of that process happening with life, with living cells?
yeah,
Where
so
is
you have
it
the so you have the factory building that is analogous to the cell membrane. You have
So we're
computers
talking
in a
about
factory.
factors. I'm asking you for evidence of how life came to be. Okay, where basically is the evidence of that's how life came to be? Okay, that somebody sat down and said, "Oh, we need a cell membrane here, and we need to have this protein machine, and we have to make this, and we have to go and order this, and we have to put that together." Where's your evidence for that?
Well, I always see intelligence making blueprints and then information transfer of that blueprint to the factory, where then the machines or factories are built upon that blueprint, is always a mental process. So what is the evidence is the path to the cause. If we only have observed intelligence being capable to instantiate that process from a blueprint or instructional information to the 3D device, which is equivalent to the information, to the digital information, then this is evidence that a mind has instantiated both for a specific purpose. And that is what we see in each living cell, where the genomic information is transferred through a complex machinery and then is expressed, and the result is the next same thing, the self-replication of the cell.
Okay, the only evidence I see here is evidence that you've taken your analogy too far, and you can't provide me any evidence that there was something mental going on when life was created, and you can't show me how that happened. Okay,
that's correct. I cannot show it to you.
You cannot show it to me. Okay, you have no evidence of basically a mental process. Like, if you look at an anthill or a termite mound, or one of those complicated little structures, okay, was that a mental process that created that?
What?
Was it a mental process when the ants created an anthill? Was that a mental process?
That's a good question.
Well, I think that ants, they have a pre-programmed program to do that.
What
is like it
is
is like
it a mental process?
Give me let me give you an analogy here. If you see a robot,
another analogy. I don't want an analogy. I want you to talk about the ants, basically.
Yeah, I already answered you. I think that ants are pre-programmed to operate and make what they do.
What's your evidence?
What's your evidence for that?
Analogously to a robot, which is also pre-programmed to do what it does.
Okay, but is it a mental process when they transfer that information from their pre-program to the anthill? Is that a mental process?
Well, the ant probably has instinct. It is not the mental.
What is an instinct? What do you mean an instinct? What are you talking about?
Otangelo, no offense, but I don't think you've seen how ants or termites build anthills / termite mounds.
Yeah. Anyway,
not how they build them. But anyway,
I kind of want to wrap this up, Otangelo.
Should I change the topic I want to talk about?
Yeah, I do.
Okay.
I would like that.
All right. Sorry, folks. I might be going a little nerd here. So, Otangelo, watched the discussion you had with LPP and Bubblegum, which was pretty hilarious. You guys were, well, I won't get into that, but basically, you made a claim in that video that I would like to discuss with you, let's just say. So I assume you've read the kind of article by Eric Lerner that started this whole mess about the JWST supposedly showing the Big Bang is wrong and utter nonsense like that.
He obviously misused the scientific paper, which wasn't at all about the Big Bang, right?
Right. Okay. I appreciate you admitting that. Or would you also, I guess, be able to admit, because the paper that you cited, the panic paper, wasn't about Big Bang cosmology
Right,
or
right.
panicking at all.
I've never claimed it was about the Big Bang.
Right, but you claimed it was cosmologists were in a panic, basically, which wasn't what the title of the paper was about.
Yes, but what is the reason? And I think I was pretty clear and
Well,
outlined
the
it in
reason
the
doesn't
stream of
matter. The
LPP.
title of the paper and the paper itself wasn't about cosmologists panicking. Will you admit that at least? It was
Well,
a
I
a
didn't
joke, a pun.
actually read. Yeah, I didn't read the entire paper to say where they outline what exactly it is that they panicked. But if you see
That's the point.
the
They're not panicking about anything. I'm talking about the paper overall in the title of the paper. The title of the paper is Panic at the Discs, and
Yeah.
some other information to go along with it. That's a pun. It's a joke. It's about Panic at the Discos. It's not about cosmologists panicking at all. So will
well,
you admit
if
that you were
yeah,
wrong on that?
sure. I mean, I mean, I have no problem if they are not panicking, but I
Okay,
think
good. So you admit that cosmologists aren't panicking on this topic from that
yeah,
paper.
I'm fine with that. Sure,
Okay.
no
Okay.
problem.
Thank you.
Okay. Did you want to
So,
address Cell Cordova's video?
I guess we can.
Yeah. So, did you have timestamps?
Yeah. Let me look at my paper here. I guess should we just go in order, basically?
Sure. It's a lot easier when you provide me with the timestamps beforehand, but that's just me complaining. I'm just a crotchety old man. I'm gonna.
Or I have them written down here, so you can just.
Okay.
Or sorry, did you want me to? You want me to
No,
send it before?
Yeah, next time.
I have a quick question, Speed. Speed. At what age did you become crotchety?
When I was, I think my mom said I was like 2.5.
Okay, yeah. So I don't think the old man part has anything to do with the crotchety part.
Yeah. Okay.
Just letting you know. Just raising the question because I'm a philosophical, deep thinking guy.
I want to point out something I saw on Ger's streams a little while ago. By the way, I linked Ger's recent stream that he just did. It's a very, very good stream. Everybody should go watch it. But you will get cancer if you watch it. But do anyway. I wanted to point out something that was said about old people are not used to LGBTQ, and they're going to die soon and whatever. No, old isn't an excuse for being homophobic and for being intolerant of other people. Old is not a fucking excuse. It isn't about old people. Young people have it too. Everyone has it. It's not an excuse to just be an old bastard who can't get your mind around what's going on in today's culture. Anyway, end of diatribe. Well, give me a timestamp.
So, real quick question again, Speed.
You interrupt.
Before I have to go.
Oh yeah.
Okay.
Why are we looking at Sal Cordova doing his Bill Cosby impression?
That does look like Bill Cosby. Oh my God. With the eyes up. Yeah, the whole thing. Anyway, I don't know. Because I'm waiting for Andrew to give me a timestamp.
Oh yeah. Let's see.
You gotta leave now, Scott.
Yeah, here in a couple of minutes, and let me give you my theory of old age and everything. There comes a point in one's life where you give less of a fuck than before.
Yeah.
For whatever reason, some call it don't give a fuck. Some people,
No fucks to give.
Yeah, but what that does is it takes the part of you that has been holding back the negative stuff in you, and it can cause you to just reveal who you are on the inside.
That's a fact.
And it's not necessarily true, but that can happen. It doesn't necessarily happen. That can happen. I think I've got I'm approaching the don't give a fuck phase of life, but it's not making me meaner. It's making me want to hug more people and enjoy the I'll say the finer things of life, but by that I mean the things that build up interpersonal relationships and the sense of mutual humanity. But I don't have a desire to unleash my anger on those young whippersnappers or those immigrants or whatever. So I think it's about not having fucks to give, but the negative stuff is a reflection of one's character.
Scott, maybe you can help me. I forgot Bree's email. What does it start with? It's some kind of critter, some kind of monster, some kind of horrible thing. Creepy? What?
Well, Bree is. Oh, I don't know. I've used the old Brian Stevens one, but it changed or whatever.
Yeah, it's creepy critters, creepy crawlies.
Here, let me pull that. You want me to send him a link? Is that what you're asking for?
That's what I'm asking you to do.
I'll send him a link, and Bree, I don't know what it's gonna say.
Yeah. Anyway.
Yeah,
okay.
X Creeper. Oh, never mind. Just the whole thing out.
Why does it start with an X, Bree? Why?
He used to be a creeper.
Why not just CR? Because I remember that part. Okay, I got it, Scott. I got it.
Well, I've just sent it. So somehow or another, he's going to get a
number.
Get a link.
Okay. Hi, Andrew.
But now I have to go. Everybody, it's been a great week. I appreciate all of you. Thanks for stopping by.
Yes.
Otangelo, Andrew, Speed, y'all have a great weekend.
Yes, you too, Scott. See ya.
Bye-bye.
Okay. So.
All right. First kind of bit of it is him just ranting about unrelated stuff. So first kind of juicy one is let's see. Let's go about 14 minutes.
About a boot? Are you saying a boot?
A boot.
Okay. Got it.
Okay. Yeah, just let it play from there.
Okay.
A billion years old and had an abundance of elements the theory suggested should have taken 5 billion years to build up. Yet this multitude of mature galaxies with smooth disks and neat spiral arms is only 235 to 500 million years out from the beginning of it all, the Big Bang event. All the new data flies in the face of Big Bang.
All right. So first of all, these galaxies aren't mature in or fully developed in any sense. Even from the panic paper, the original paper this supposedly came from these discovery of very early disk galaxies. They define disk galaxies and spiral galaxies, and they're actually not synonymous with each other. You can have a disk galaxy, as far as I can tell, without spiral arms. So just because they found these early disk galaxies does not mean that they have these neat spiral arms that they claim in this article here. Hi, Bree.
Hey. I'm in. Let's hear it.
So the other thing I want to say was
first of all, what article are we looking at here? Are we looking at Lerner's article?
I don't think so, because he linked it in the description, but I can't access it for it's a Telegram, I think, and I have no idea how to access it. So
was that the Behold Israel?
Yeah, I think so. Yeah. It's the first one he linked, I think.
Okay.
Anyway, just kind of to wrap up my point here. The other reason they aren't mature, and one reason they aren't mature that they actually admit in this panic paper. Later on, they say that the early disk galaxies they found, some of them had a propensity, was the word they used, for tidal features and clumpy regions, and this distinguishes them from Z equals 0, in other words, redshift 0 or nearby galaxies. So this claim that they're fully mature and that they're the exact same as nearby galaxies is just wrong. There are many distinguishing features between very, very distant galaxies and very local galaxies. So that's the first point I want to make.
Okay. What do you want me to do?
I don't know. Did you look like you wanted to say something, Otangelo? Did you want to say something?
Yes, I quoted this at LPP. I can quote it here again. So this is the problem here. Big Bang cosmology predicts that galaxies evolve over long periods of time. If
Correct.
galaxies were all formed long ago, distant galaxies should look younger than those nearby, because light from them requires a longer time to reach us. Such
Correct.
galaxies should contain more short-lived stars and more gas, out of which future generations of stars will form.
And they
And
do.
in contrast, the Genesis model, where God stretched out the heavens and created a mature universe, in the same sense as he created Adam looking mature and grown up, even after being created instants ago, predicts ensembles of galaxies close to us should look statistically the same as those far away. And that is what is being observed through the new J-Webb telescope.
Nope. You're wrong. And I just gave a reason. Well, one reason why. The other thing is, have you heard of the Lyman alpha forest, Otangelo?
No.
Okay, so basically it's this pattern of absorption lines in the spectra of these what are called distant quasars. And you can tell from that at higher and higher redshifts, the gas content of these galaxies increases. That's consistent with the Big Bang model, where things are very close together and not all the gas has been used up to make new stars. That's another reason why these very early galaxies are not the same as today's nearby galaxies. So I don't know where you're getting this information from, but no, the James Webb is not showing fully mature, whatever that means, galaxies.
I think this is precisely the shocking thing here, that they were not expecting mature galaxies fully formed as closer to us.
That's when they're not fully mature. They're not fully formed. Where's your evidence for that?
I have many articles I can give. I mean, galaxies in the infant universe were surprisingly mature. This is from Science Daily. I have many references. I can put the quote here.
From all these news articles? Where's your
I have
paper?
Not only news articles, but also science papers. This finding contravenes current models underlying added for that period of cosmic evolution. This is a science paper from 2019. The existence of these large number of massive and dusty galaxies is unexpected in current models of simulation.
Was that correct? Yes, 2019? That's before James Webb was launched.
Yes, they have seen these already prior to the James Webb.
But that means James Webb isn't seeing these new galaxies that.
Yeah, that was the original claim, Otangelo, was that James Webb is revealing new information that
which I mean, you are correct. We do observe some of these early massive galaxies, but have you considered at all, Otangelo, that one of the reasons we sent James Webb up there was to update our pretty uncertain models of galaxy evolution? Because I don't know if you noticed, galaxies aren't simple rotating disks with stars in them. They're very complicated objects, and we need to better understand how they mature over time and in what ways they originally formed. That's one of the reasons, or one of the main reasons, James Webb was launched to begin with. And it's doing that spectacularly so far.
Yes, I agree with you, but nobody said that what is being outlined and exacerbated now with the new data of the James Webb wasn't already something observed prior to it, and which was putting a big question mark to the Big Bang cosmology models.
So now you're claiming these this is a problem for Big Bang cosmology?
Absolutely.
Oh, because I thought earlier you were
Absolutely.
claiming that this wasn't from
No,
that
I've
paper.
never said that.
Okay,
No, I've
okay.
never
So
said that.
why do you think it's a problem for Big Bang cosmology?
Because fully developed and mature galaxies shouldn't be found so close to the Big Bang. They should be much much younger.
They're not as close as you think they are, and they're not as fully mature as you think they are. I just gave reasons why. Here's another one from the paper, the Panic paper itself. They measured distances of greater than 3 to around redshift of 6 or 8 in some cases, but that's not 500 to 250 million years after the Big Bang like you claim it is. It's lower. It's slightly lower than that, but it's still several hundred million years to up to a billion years after the Big Bang. Now, granted that some of these results are earlier than we previously thought, based on Hubble Space Telescope observations. But Hubble Space Telescope observations are not the same thing as what was predicted by the Big Bang model itself. Do you understand
Well,
that?
I'm just reading here. 2 other groups claimed an even older galaxy, which could have existed 225 million years after the Big Bang.
What
And the
what
third
morphology
group,
did they claim it was?
and the third group is claiming to have observed a galaxy that was formed at the staggeringly early time of 180 million years after the Big Bang. That's where the trouble comes in. So
Yes,
I can
and
I can
what
I,
shape did they claim they have?
well, I can link the paper and you can look it up.
Oh,
I mean,
I don't want
these are not my
you're
claims.
making.
These are not my claims, Andrew. This is something which others are observing and pointing out.
Finding these very distant galaxies at this particular time is not the same thing as finding fully mature galaxies at this particular time. And in order to demonstrate that, one, you need to give me the citation and the evidence that shows that these these two galaxies, candidates, I should note, at these very early times, are in fact the same as the ones showed in the Panic paper, which are these slightly more mature disk galaxy shapes. I don't think you can extrapolate one from the other.
Yeah, I mean,
Can I just real quick, like I say something about this whole topic real quick?
Sure.
You all know animals are more my thing than talking about stars
go
and
ahead.
stellar formation. But if we have this new technology and we discover these or make these new discoveries, which cause us to rethink our model, our Big Bang model, or how we view redshift or any of this new information, changing the way we do it. That's not a bad thing, and it doesn't disprove the things that we've already established regarding
Right.
the current model. And it's just it's more information to it. There's more subtlety to it, especially with what I'm understanding. And correct me if I'm wrong, but that what we are understanding, the new information that we have gotten from the James Webb satellite is comparable to what we have collected in the 50 years before that, just in these first few months. Is that accurate?
Yeah, I don't know the exact. Yeah, that sounds about probably right. But yeah, that's a good point you're making. And that's a point I really want to drive home here, is the fact that if we do in fact find out that these candidate galaxies, and I do want to note that the issue isn't settled, because for one thing, I don't know if you noticed, Otangelo, the James Webb hasn't been fully calibrated yet. The redshift distances that they're getting, or the redshift values they're getting here that correspond to a particular distance, those are photometric. So they're about the overall shape of the light curve. What needs to be done is there need to be analyses taken of its spectra, of the individual absorption lines. That will tell us the proper actual redshift and therefore its distance.
Yeah, Do you happen to know what our error margins are right now on that?
No,
And-
but it's pretty uncertain still.
Okay.
And
Of the data that we've
people in the papers are even mentioning that. Sorry, go ahead, Bree.
of the data that we've gotten back, how much of it has actually been analyzed, and how long was it going to take to analyze the current? I mean, because I would imagine we've got a lot of numbers.
So the data that has been sent out right now is preprint, so it hasn't been peer reviewed. I think they are pretty confident in it, but I think there are some articles that have also shown that even some of the supposed high redshift galaxies they've detected with James Webb, some of those have already been shown, oh, this was just dust that made the light appear red, but it wasn't actually red shifted out to this particular distance. So we have to be careful with what we're calling settled science, so to speak, or what are these high redshift candidate galaxies, and what will we eventually find to be actual high redshift galaxies, if that makes so. It's not terrible, but it needs a lot more confirmation for us to be sure, and that's the point that some of these people who wrote these news articles and Learner's article, for that matter, haven't quite grasped yet.
Yeah. And my understanding was that what it affected is their models of early galaxy formation.
Correct.
And that was
no
something.
way disproves the Big Bang.
Yeah, and it was something that we didn't have a lot of information about before.
Right.
So now,
Yeah, exactly.
we have a lot more to draw from. So we had models, various hypotheses, but now we have data. So.
Right. Because think about it. You had the Hubble Space Telescope, could see out to a particular distance and particular range of wavelengths of the light picking up. Now, it turns out different wavelengths of light will actually tell you different types of information about a particular object, and galaxies in particular, because in one wavelength, you'll see a lot of activity, a lot of stars forming. In a different wavelength, you'll see less activity because the stars are different. But if you take that into account, the James Webb has a much deeper reach, like you said, and can probe these much longer wavelengths, the infrared and deep infrared, whereas the Hubble was more about the visible and ultraviolet part of the spectrum. So of course you're going to see different things from these different instruments with different sensitivities, and like you said, it's telling us new exciting things about what we thought was true based on the Hubble results at a particular sensitivity, and now that we've expanded that sensitivity to a different range, we're finding new things. Of course we're going to see that. Like this is what we were hoping for. This is why we built the telescope to begin with. I think you're muted, Otangelo.
Do you have another bookmark, by the way?
Okay.
Or
so let me quote here. Here another article from June 19th. Once again, galaxies look surprisingly mature shortly after the Big Bang of the universe. So very clear.
Okay, where's that? Give me the context there.
Are we looking at pop science sources and
right.
that are all
I don't want you to give me
the
pop science sources. I want you to give me actual papers, Otangelo.
Yeah, this is from universetoday.com, and the title is, once again, galaxies look surprisingly mature shortly after the Big Bang of the universe.
Is this from the same press release as the last one that you were reading now? The one that I think the one you linked from Big Think? It's the same press release, right?
No, I think not. I just researched this right now on Google.
okay,
what's the date when this was written? Just curious.
19th of June 2022.
Okay.
So we got it on the screen here. Let me put that in the description now that we mentioned it.
Yep. Did you say you wanted another timestamp, or you want to look at this?
I'm going to put it in chat. Well, yeah, we
Okay.
can look at this too.
Okay.
I put it in public chat, and I also linked it in the description. Okay, a young galaxy with a catchy, roll off the tongue name.
Yeah.
A
Sorry, I think we're gonna need to do a separate thing on the south. We can just go over some other stuff right now.
Okay, good.
We'll
Yep.
do
Smaller
a
stuff.
whole rant about that. So stay tuned.
You got a
Yeah,
lot to
let's,
say about the South stuff. Are you doing the ranting thing on Saturday night again,
Maybe.
Andrew?
Or I should be free.
Yeah.
If you're okay with that speed.
yeah.
Okay.
Yeah.
I might
Saturday.
come on there with you if I'm free.
I think Saturday is good. Sunday may not be for me today.
Okay. We'll figure something out Saturday then. Okay, Bree?
Okay.
Sounds good.
Would have looked just 700 million years after the Big Bang is larger than initially believed, with significant outflows of hot gas from its core, halo of cold gas emanating from its outer rim. Let's see, it's considered representative of young normal galaxies as opposed to massive galaxies. And new observations suggest that the adolescence of normal galaxies may be more rambunctious than previous models suggest. Okay, so more rambunctious than previous models suggest. What part did you read, Otangelo? You're muted, Otangelo.
No, I just did read the title of the paper.
Oh, okay. Well, yeah. Well, we got to go a little deeper than that. Okay, so more rambunctious than previous models suggest. So what they're indicating here is that they're going to have to modify their models, right?
Yeah,
So
that's
why is this a
what
big
it's all
problem?
about. Well, because
Yeah.
the
And
models
why is it a problem?
they have to be rewritten. I mean, I think the deeper implications that
Now I can read it
take
too.
time.
So how do you get from that to a problem for the Big Bang?
Right.
I don't
Well,
see it.
the Big Bang Theory has to be rewritten because we are not
no.
talking about the
Where are you
beginning
getting
of the
that
universe,
from?
which is
Where'd
You
you
don't
get
throw the baby out with the bathwater when new information comes in. You incorporate it into the existing theory.
Yeah, I mean, how do you get to that, Otangelo? I mean, you're making and why are you making that leap? Do you think this has something to do with God?
absolutely.
Really? So,
Yeah.
okay, so if the universe wasn't created, basically, like at some instant, if it's always been here, because if we tossed out the Big Bang, what we're going to end up with is probably an infinite universe. That would be better for God. How would that be better for God?
No, nobody is doubting in regards of the beginning of the universe, which is why
like a
when
Big
the Big Bang model was actually brought to the spotlight, they all tried to deny it, and they preferred a steady state model because then you don't have a beginning and you don't have to explain the beginning. So
Right. So,
this is
so how,
about
how is
the
this
beginning.
helping you? How is this helping you that we have to modify our models so that they're more rambunctious? So how does that?
I still want to see how they will actually modify the models in order to be consistent with the naturalistic evolutionary Big Bang model. I still want to see that. I don't see. I think this will be one of the many open questions which just will float there as open questions, and that's it. Now, I as a creationist, of course, I have a case because I can say God created the galaxies right at the beginning, fully developed. They seem
they're not fully developed.
And that conveys with Genesis.
I'm still waiting for your evidence that they're fully developed, Otangelo. Where are you getting that from? Where's your evidence for that?
The title of the article here says it. They are mature.
Yeah, the title tells me nothing of substance.
Well, what does mature mean? For me, it means fully developed. Maybe I am wrong about that.
You think if I find a random article, the title will tell me everything I need to know about the information in that article?
So
or do you
maybe
think
we should still wait a little bit more.
Catchy title?
Okay, Otangelo, let's just admit it out front that this gives you hope. that they're going to discover something that proves God, right?
No, I've never said that I expect to prove God. Of course,
But
we'll never do that.
This
What we
gives
can
you,
do is,
right? So like you're hopeful. You saw this title and you got hopeful. Like you, just like Cell did. He saw the title Panic and he just went off the rails with hope that somehow science was going to support the universe coming into existence all at once 6,000 years ago. You have hope.
yeah, I don't say actually speed. I don't actually say it was 6,000 years ago. I could be fine with 14 billion years and still believe in a young Earth creation model.
Okay.
So it looks like, from my understanding, that the term mature galaxy is kind of a buzzword.
Surprisingly
It is kind of it depends who you ask, because in some ways they could be considered mature, but in other ways they're vastly different from the ones we see today.
Well, I've talked to some.
That's what I mean. Like, what do you mean by mature in this case?
Yeah, I've talked to some 8-year-olds and come away saying he was surprisingly mature.
I'm seeing everything from 2 to 3.5 billion years to be mature, considered mature. So it's what is a mature galaxy? What are you inferring that as, Otangelo?
He means completely formed, and I don't think that's what they're seeing, right? Isn't that the case, Andrew, that these things are still, somewhat immature?
Well, for one thing, they're all... Sorry, go ahead.
Go ahead. Oh, I was going to say, saying they're fully matured. What does that even mean? I thought that galaxies just went through a cycle, that they have stages that they go through in their development towards wherever.
Kind of, they have there are different pathways. As like I said, our models of galaxy evolution are still a bit uncertain, but there are general pathways we think they go through, depending on like the circumstances of how many galactic collisions they have. If they have a bunch of big ones, then they'll probably become a large elliptical galaxy. For example,
depending on what type of galaxy it is, depending on what type it is, does that change how fast it matures?
It could definitely. That's what we're trying to figure out right now, is how rapidly this occurs for different types of galaxies.
So it sounds like the term mature galaxy.
That's actually one of the things they. Sorry, go ahead.
It sounds like the term mature galaxy is not something that we could even demonstrate at this point with the information that we have.
Right. Yeah. Exactly. It's for one thing relative, and by different metrics, we're not certain of that at all right now, is what I'd say.
Yeah.
And I think most astronomers would admit, based on the limited or the still growing information we have currently.
Yeah, and this was with a different telescope, I think, that they observed this.
Well, I still hadn't had Otangelo answer. What do you mean when you say mature galaxy? What does that mean to you?
Fully developed.
Just a stage of its development. Okay. Yeah. So not so much a time frame then, but a stage of development. Time is different depending on what type of galaxy it is. Okay.
I'm just saying that there's nothing wrong with having hope, Otangelo. You can have all.
Thank you, Mike. You're so nice.
But it just doesn't mean anything to us that you have hope. That somehow the James Webb is going to prove that the universe is older than we thought. I don't know. More mature than we thought.
Came into find God on it. We could find God.
Yeah, like we're not going to find any evidence of galaxies forming. Do we have? So, Andrew, can you respond to that? Do we have evidence of galaxies, like earlier, younger galaxies, anywhere?
Oh yeah, definitely. I don't know if we're quite at the point yet where we're seeing like the actual like embryos of galaxies, or the first stars for that matter. I don't think they've reached that stage quite yet. Hopefully they will. But they're definitely seeing these very young galaxies in very early stages of development. And like I said, it's relative. But for one thing, a point that's often missed is when you look at the papers that they're citing here, or not Universe Today, but these Eric Lerner and other people like him, actually take a look at those papers, and most of them find, oh yeah, these galaxies, they're a lot smaller than the ones we see today. They're compact is the term they use. I think one was they found size ranges around 5 kiloparsecs, which is around 1 to 2,000 light years, give or take. So it's very, very small compared to the galaxies we have today, like the Milky Way, which is around 100,000 light years across end to end. So these are very tiny galaxies, and we can tell from that point alone, as well as some other things, that galaxies have changed over time, because the galaxies back then are not the same as the ones we see today. Definitely.
So the problem
So this is an interesting article here from creation.com from 2004. Okay.
No.
And they are already back then, 18 years ago, pointing to that thing of mature galaxies. And in the end, they write this here. The existence of such large, mature galaxies lined up in a beautiful filament makes more sense when interpreted within the biblical belief system, that this God rapidly
why should
and
I
supernaturally
there?
created fully formed stars and galaxies on day 4 of the creation week.
Well, you dove way back for that one, 2004, huh?
Yeah.
This article here, yeah, okay. Young galaxies too old.
What are they saying here?
I don't know. Let's see. Let me put that in the description before I forget.
Can you summarize the evidence they have, Otangelo?
Okay,
The
4
same
meter.
that we are the same evidence that we are observing now, only that then they said the galaxies were 10.8 billion light years away, while now they are basically very close to the Big Bang, just 225 million years after the Big Bang. So we are getting closer and closer to the Big Bang event.
Right, that's what we're trying to do.
Yeah. Okay. So anyway, this one was 10.8 billion light years. So the problem seems to be carbon gas, and carbon gas, if I'm not wrong, was from supernovas, right?
Yeah, probably a
That's
large portion of it. Yeah,
yeah,
one
but then,
way. Yeah.
Yeah, that's one theory, but there's other possibilities here because I think there's some issues with that. Other ways for it to form. However, the discovery is a huge problem for evolutionary timescales. Okay, this has nothing to do with evolution. This is cosmology. What in the hell? Why do they do that?
No, they are talking about cosmological evolution. These are
that's a thing. I think it's just not the same thing. Like some people
don't like
it to be.
I don't like it.
37 mature, bright galaxies lined up.
I mean, Lee Smolin even talks about astronomical natural selection.
Yeah, it sounds like.
So are they talking about like an early proto galaxy cluster here? Because that would make sense in context. What are they saying?
10.8 billion light years away, so that was 4 billion years after.
They have to give the specific redshift there, because distance is kind of it gets a bit confusing on these scales.
Yeah.
It doesn't necessarily correlate with the actual age.
Let's see if
it takes too long to explain.
Okay, let's see. Here we go. So here are the sources.
Okay, it was a galaxy cluster.
Yeah. Oh my God, that's from creation. I don't want that one. Okay, so one is. Okay, so I'll copy those two into the description just for the hell of it. Oh, anyway.
Okay. I do want to note what Dave just pointed out. He is correct. when they do calibrate it, like people are going to be flying over this thing because it is a valuable new tool at our possession. And I don't know how many people notice, but like telescopes, they're not free to use for everyone. Like you have to schedule, okay, when are you gonna be using it to look at a particular patch of the sky, how long you're gonna stare at that particular patch of the sky, and what objects you're gonna look for. So it's people are kind of waiting in line for James Webb, definitely.
Yep. Yes.
This is a several billion-dollar telescope to make, so of course we're going to use it all we can.
I'm going to figure out a lot of things.
Yep.
And if the Big Bang did go away, I'd be like thrilled, but I don't think it's going to.
Who knows? It might. Doesn't seem likely at this point, though.
Nope.
Extremely unlikely.
Okay, what else we want to do, boys and girls, before we shoot it in the head? It's 5:30, 5:35.
Oh wow, time flies by fast.
Yeah, it does. Yep.
Well, I want to say, Otangelo, thank you for discussing this. I obviously disagree with most of what you say, but I appreciate your courage coming in here to talk about this.
Yeah, and your absolute hope, in spite of like no evidence for so long, just inspires me, Otangelo. You inspire me.
we agree to disagree.
Yeah.
Okay.
And also,
Okay.
Check out my new club moss. It just arrived in the mail. One
club moss.
One of my patrons bought it. I'm trying to light on.
Is it alive?
Yeah.
Oh no. Oh no.
Club moss.
Oh no. Yeah, but do you sleep with that around? Because I don't know, I've heard bad things.
Oh, yeah.
Yeah, it's beautiful. Beautiful.
Isn't that gorgeous?
Hang on a second. Let me look.
That's nice.
I love club mosses.
God.
They're like my favorite
Ooh,
plants.
that's a good specimen.
Yeah. That we used to call that Christmas pine, and my uncle used to make us pick it for Christmas wreaths, and he'd pay us like 25 cents for a stuffed full gunny sack full of it. Then he'd make $5 Christmas wreaths.
Like it's getting ready to.
Yeah, nice.
Too. Yeah.
And I always find club mosses and other lycopods so interesting because they're vascular plants, but they're not quite ferns. That kind of level of complexity, so they're pretty interesting. And the fact that in the Carboniferous they were or one of the dominant groups of plants.
Yeah, they were the trees of that time period. The thing about it is that people always think of trees as like it's not a clade. Tree is a niche. It's a biome to be filled. It's not an individual organism. But this is a they call it a spike fern or spike moss. This one's not the peacock like what I used to have, but it's pretty close species. Which one is this? This one is Selaginella crossian. So this one's going to stay green. I have another one at my other house that's rainbow colored though. It's gorgeous.
Do they do well in in the heat? Like are they heat intolerant?
They do all right as long as they're the main thing that they need that these ones need
probably moisture
is to be in shade and kept wet. Yeah.
Okay. Because I got a lot of shade here, but I'd like so you can buy live moss like on Amazon and stuff. Or
yep.
Oh, cool.
People send it to me.
I would like moss.
I can't buy bugs on Amazon, so people send me plants instead.
Yeah.
I once took a little patch of moss from a park nearby and I put it in a little jar. I think I probably killed it.
Well, I do that and put it in water and put it on my microscope and keep it and then I forget about it and I look at it and I go, what is this dried up piece of dirt I got here? It's like, oh shit, that was my moss. I forgot to water it.
Yeah, I don't find all botanical topics interesting, but diversity and classification, that's interesting to me.
Especially when you get into mosses and these old timey. These used to be the kings right here. Before vascular plants took over. Yeah. All right, that's all I wanted to go over. So I just wanted to
Thank
show
you, Bree.
stuff off.
I gotta go get
What's
something.
this? I've never seen a live club moss in person before. Haha.
Yeah, I've seen them in, or like Speed was talking about, as wreaths and stuff, but I'd never
Yeah.
really had one.
I've seen like the pictures of horsetails and stuff like that, but never actually. Or I guess not in person in this case. You know what I mean.
I woke him up. I woke Snack up. There he is. Oh, the lighting is bad for Snack. Hey, Snack, what's happening? And this guy is a terrorist. He, like, if I open the door, he runs just straight outside like a flash. And if I happen to open the door and close it, he smacks right into the door.
Oh no.
Yeah.
What's he doing to get out there?
Oh, he just runs around like crazy. I've got a bunch of jewelweed growing. It's oh my god, I went out of focus. So jewelweed is they make really beautiful pitcher like yellow flowers, and then when they form fruits, if you touch the fruit, they blow up in your hand. So we call them snap beans. But anyway, I've got a forest of that, and it's got this really thick stalks, and then it grows about chest high. And he runs around out there and that, and he stalks the other cats, and he terrorizes them. And he's a terrorist. He's constantly biting the other cats' tails, and they won't defend themselves. They just growl at him, and he's not impressed. He doesn't give a shit.
Yeah, same thing with my cat and my dogs.
Yeah.
Well, or at least used to be the case. Not so much anymore, thank goodness.
They need to beat him up once, and I didn't realize my cats were so peace-loving and docile until I got
cats.
Yeah,
they're Buddhist.
Cat.
Your cats you raised Buddhist cats.
I know. Anyway, we're going to shoot her in the head, guys.
Spooky.
Okay. Anyway,
night, everybody.
Thank you, everyone. Bye.
We had like 20 watching at one time. Otangelo, you bring them in. So, thank you, Otangelo. Good talk. And
take care.
goodbye, cruel world.